Good morning, everyone. Welcome to the 2026 Royal Tyrrell Museum Speaker Series. My name is Dr. Christopher West. I'm the curator of paleobotany here, and I'll be your host for today. On behalf of the museum, we would like to thank the Royal Tyrrell Museum Cooperating Society for their generous and ongoing commitment to the speaker series. Our work takes place throughout the province that is known as Alberta, and we gratefully recognize and honor Indigenous peoples' long-standing connection to these lands. Today, we welcome Dr. Don Brinkman, who is our speaker today. In 1982, Donald Brinkman began working for the Royal Tyrrell Museum as curator of vertebrates. Later, he was then the director of preservation and research for a decade until his retirement in November 2018. Dr. Brinkman continues to study how ancient animals lived in their environments during the age of dinosaurs, with a focus on turtles and fishes. He is interested in patterns of distribution and how they relate to environments and climate. So everybody, join me in welcoming Dr. Don Brinkman to the stage.
So thanks everybody for coming. Yeah, I just want to start by recognizing some of the colleagues who we've worked with on this project from various institutions and give you a bit of an outline of what I plan to do. I'll take about the first half of this talk to give a background, to provide the context for interpreting the significance of the assemblage from Uzbekistan, and then go through the fish from Uzbekistan and end off by looking at distributions and what that says about patterns of dispersal. And I want to go back for an introduction. I want to go back to the beginning of the museum. So when the museum was being envisioned back in the early 80s, the decision was made to make the focus of the museum paleoecological. This was pretty innovative at the time, because at the time, paleoecology was viewed as a very soft science. It was basically storytelling. And the rigorous paleontology at the time was really focused on cladistics and working out interrelationships. But there were things of paleoecology that everybody agreed was significant and could be studied. And one of these was the question of interchange and connections between Asia and North America. And the poster child for this was this dinosaur, Saurolophus, which was found different species, but clear the same genus in both Asia, in Mongolia, and in North America. So this provided clear evidence for some kind of connection between these regions. And because of this interest, the early planners of the museum, Bill Curry and colleagues, had big ideas, and they wanted an international project. I asked him how it came to develop. He said initially they were looking at working with Mongolia, but through a series of just lucky circumstances, they ended up with the Canada-China Dinosaur Project. And the goal of the project was really to explore this idea of connections and similarity in faunas between Asia and North America. Unfortunately for me, the approach was to look at the total fauna, not just the dinosaurs. I was interested in turtles and involved in looking at turtles. And through this project and a number of other researchers looking at these questions, we came up with a very clear understanding of dispersal patterns. So there's a number of groups that had originated in Asia and moved into North America at various times. The trionychids are widespread in Asia in the early Cretaceous. They first occur in North America in the Cenomanian at the base of the Late Cretaceous. Macrobenids, Zodokids, Nanchunchelids appear in the Turonian about 90 million years ago. And testudinoids, which are a dominant group in Asia in the Late Cretaceous, first appear in North America in the Eocene. So there is a clear pattern of dispersal that's actually in line with Ian's talk last week. But for dinosaurs, things were a little more complicated. And part of the reason was that there's big ecological changes. So assemblages in North America are preserved in very fluvial, wet environments along the coast, whereas the localities we're looking at in Asia were arid and sometimes full desert environments. But early in the project, in 1990, the other aspect of this is that there's a number of groups in of turtles in North America that are restricted to North America during the Cretaceous. Again, part of the pattern Ian presented. But again, we had this ecological pattern for dinosaurs. So early in 1990, we were visited by Lev Nesov, who was then from Leningrad, now St. Petersburg. And he had been working in Uzbekistan, which is on the edge of the Asian continent, in the red dot in the your lower right here, and in an environment that was very similar to Dinosaur Park. And he was seeing things that were looking like the kind of thing we're seeing in Dinosaur Park. And the thing that really impressed me was a fish represented by scales. At the time, we didn't know what this fish was. It didn't have a name. So I was calling it Holosteinae. In a project led by Jim Gardner recently, we found that we're able to show that it had been named by Cope, and the name is Arotus hieroglyphus. We still don't know what the fish is, but at least now it's got a name. So, and this was so similar that if you put the Asian specimens in with the North American specimens, you couldn't separate it morphologically. So clearly indicating some kind of connection with the fish. So there's a story about fish interconnections as well as with the turtles. So Nesov had been working at a number of localities, and two are particularly significant. One is Ecological, which is in the Cenomanian. And there's two terms I'll be using quite a bit throughout this talk. And one is Cenomanian, which is the very earliest time period of the Late Cretaceous, from about 93 to 100 million years ago. And the other is the Turonian, from about 90 to 93 million years ago. Between these times, there's a period of high global temperature, which seems to have an impact on faunas. And that's part of what we're looking at when we're looking at the turtles and the fish. So he had been looking at these two and had gotten good assemblages from each of them, and was able to put together pretty extensive faunals from each of these time periods. This area of interest led to an exchange called the Uzbek, Russian, British, American, Canadian expeditions, called the URBAC expeditions for short, led by David Archibald, Alexander Avrianov, and Hans-Dieter Seuss. And again, like the Canada Dinosaur Project, they were attempting to look at the whole faunas. And I just put some references up to give some sense of the scope of the taxonomic diversity they were looking at. But the one group that they weren't able to look at was the fish. So there is one paper come out on one of the fish, but no real understanding of the full diversity. And the reason for this is because looking at isolated specimens of fish from the late Cretaceous fluvial beds is challenging. Work on fossil fish involves looking at articulated specimens, whereas in fluvial beds, what you're dealing with almost exclusively are isolated elements. I had started looking at fish based on microvertebrate assemblages from North America. I was seeing distinctive morphological groups that had a distinctive pattern, and I wanted to quantify this. So I adopted just a strictly morphotype approach. If there's a morphologically distinct type of centra that I thought was taxonomic, I gave it a letter designation and treated it as an operational taxonomic unit. So when I started, and this is from one of the very early papers, there's a couple that I could recognize and put in some kind of teleos group, but the rest were all strictly morphotypes. Through comparison with recent specimens and fossil specimens, it's been able to update this and put names on most of these. There's still some that are morphologically distinct, but I can't put them in any modern group. But we're getting through this, we're getting a fairly good understanding of the diversity and the taxonomic groups. It's not possible to do phylogenetic studies with this kind of material, but they do provide information on distribution and time of occurrence, which is useful, especially for paleoecological studies. And initially I started with one locality, looking at material from southern Alberta, and was able to expand it to looking at a number of localities that covered large geographic and stratigraphic ranges, going from Utah, in southern Utah, up to a locality in Alaska, and covering through the entire late Cretaceous. And by looking at diversity in different time periods, it was possible to put a chart together showing the diversity patterns. It had already been recognized that there was a big faunal change between the Cenomanian and Turonian, And with the isolated elements from microvertebrate sites, it was possible to add to this so that what was recognized as a number of taxa that were distinctive for the early Cretaceous had either gone extinct or become very rare. And that was things like a primitive fish called Lepidotidae and another group called pycnodonts. They didn't go extinct, but they became very rare. Whereas there's a number of early first occurrences of interest. And one of these was an amiid, a large amiid called Melvius. And this was of interest biogeographically because it's a member of a family, the Vitalaamidae, that's well known in the early Cretaceous in Gondwana in the southern hemisphere. But its first occurrence in non-marine beds in the northern hemisphere is in this Turonian time. Another one in Utah, the lepidotid first occur in the Turonian. If you go to the paleobiological database, you'll see reports of lepidotid in North America in the Cenomanian and Late Cretaceous and early Cretaceous, but these are all pretty much all based on scales. And the problem with scales is that it's scales of gar, lepidotids, are virtually identical in external morphology to the scales of the lepidotids. So that reports based on scales in these early localities are not reliable. What's distinctive and unique about lepidotids is the centra and the teeth. The teeth have an infolded dentine, which gives striations on the base. There is a tooth that from the Cenomanian referred to lepidotids from Texas, but it doesn't have this striated shaft. So I don't feel that it's a reliable indication of the group. So that it's appearing that lepidotids first appear in North America in the Turonian, whereas again they've got relatives in Gondwana in the early Cretaceous. So again, looking like something that's dispersed during this Turonian from the southern hemisphere to the northern hemisphere. And the other case of this is one that came up from looking at the center of teleos. There's these really morphologically distinct teleos. I had been identifying them as a member of a group called the Otophysi, which includes things like the cyprinids, the catfish, and some other groups. We're able to confirm this from a specimen that was collected in Dry Island, actually found by Andy Newman. And so I'm anxious that he get out and spend more time there. And we're able to work out the structure of the individual elements and segment out individual centra and compare with the isolated centra so that we know that this is a member of that group. And that's significant because the group is widely recognized as having a southern hemisphere origin. So again, another group that originated southern hemisphere and appears in North America in the Turonian. So this was what I was familiar with, the record of fossil fish in North America. Lev Nesov summarized his work in a publication in 1997 and did a number of things. He went through localities and reported fauna at each localities. He provided just extensive illustrations. He was a master of fitting a maximum number of specimens on a single plate. Most plates have 20 to 30 specimens, and he has 60 plates in it. And the specimens he whitened before photographs, so they're pretty recognizable what they are. And with the technology these days, it was possible to use Google Translator to go and read his text, which is really an amazing thing to do when you've grown up seeing the Russian literature as something inaccessible. And when I did that, what stood out was that he was seeing some of the changes in Uzbekistan in Central Asia that were the same as we were seeing in North America. So that in his localities, Lepidotidae and pycnodonts drop out entirely at the Turonian, and lepidotids first appear. So that struck me as something worth looking into because of previously these faunal changes had been looked at as a result of a local environmental change, something like rise in sea level. But with the concurrent changes in both these regions, we're looking at something global, something significant in terms of the evolution of faunas globally. And I was able to make connections with researchers who were collecting in there and were interested in having somebody work on the fish. One of them is Martin Kundrat. Martin had been a student volunteer at the Tyrrell Museum back in the late 90s, early 2000s, and actually was supported by the Cooperating Society. So from that, support went off and did great things. So he had been part of a joint project with the Uzbekistan, looking at faunas from the same localities and beds that Nesov had looked at. And we're also joined by David Ward, who was a member of the Urbak expeditions and had specimens that were surface collected and also information on the geology that was critical. Martin, because of his time in working with the Tyrrell, was very familiar with the idea of screen washing and looking at microvertebrates as a fauna. And he had applied that approach in Uzbekistan, so had a huge sample, over 4,000 specimens of small teleocentra to look at. So that leads to the part of the talk that I'm going to give you an overview of what kind of fish are present and try and show you how much similar some of these are. So one of them that's very abundant in Uzbekistan is an amiid. Its amiids extend throughout the Late Cretaceous in both North America and in Asia. And we don't know the species level taxonomy, so we can't say anything about the relationships of the amiid from Uzbekistan and North America, but it's a significant faunal similarity. And again, the gar Lepidotidae is well represented in the Turonian localities by fully diagnostic material. And it's put in a separate species, but this is largely a reflection of geography. Belnostomus is well known here to anybody who's worked in Dinosaur Park, and it's present also in Uzbekistan. I had initially assumed that it was, it looked the same, it was the same, and then a reviewer pointed out that Nesov had given the Uzbekistan a distinct species. And when I looked at it closer, there's a clear difference. In the Uzbekistan Belnostomus, the tooth row extends out to the tip of the premaxilla. In the North American one, which is put in a different genus, Longirostris, it ends well before the end of the premaxilla. We don't have a specimen of Belnostomus with the full, complete lower jaw. So we don't know how long the premaxilla was ahead of the tooth row, but we know there was some significant length. So if anybody's out there collecting fossils in Dinosaur Park and sees a complete Belnostomus, I'd really urge you to make a special effort to collect it. Again, Arotis is one that I'd mentioned earlier. As well as occurring in Dinosaur Park, it occurs in a Turonian locality in high Arctic, in Axel Heiberg Island, and in North America extends into the Paleocene. So this is an interesting fish from its distribution and would be really nice to know what kind of fish it is. One of the differences in at least relative abundance of the Asian and North American assemblage is the presence of a ichthyodectiform. Ichthyodectiforms are best known to us by Zephactinus, and this is a picture of the specimen we have on exhibit. They're diverse in marine environments, but they also occur in non-marine environments. So this is one Iadichar that occurs in the Uzbekistan fluvial environments, both in the Cenomanian and Turonian. I've indicated its presence in North America on the basis of two specimens. There was an articulated specimen described from Texas of the Cenomanian. It's a little uncertain of the environment of deposition. It's a formation that has both brackish and freshwater beds in it. So I don't know if that specimen is North American, but it might be. So I've included it as a evidence for an ichthyodectiform in the fish assemblage of the Cenomanian in North America. And there's a specimen, this articulated tail from the Kaparwitz Formation of Utah, and that's equivalent to Dinosaur Park. It's got a pattern of ridging that matches that seen on ichthyodectiform centra that seems to be typical of ichthyodectiforms. So, and I haven't seen any other fish that has this same pattern of ridging. So if it's here, it's very rare. I've never seen centra of this kind of fish in microvertebrate assemblages. There's one other specimen in the Bissekty Formation, the Turonian of Uzbekistan, that seems to be an ichthyodectiform. I initially thought this was maybe the anterior centra of Iadichar, because centra on the anterior of the column tend to be shorter than mid and posterior. But it's different in terms of having a mid-ventral and mid-dorsal pit, so it seems to be taxonomically distinct. I've only seen this specimen in the Bissekty Formation in the Turonian. So it seems to be something that's endemic to that region. Elopomorphs are typically thought of as marine. They're full marine today, but they're really commonly present in non-marine assemblages in the Cretaceous. We're familiar in Dinosaur Park with paratarpon, often represented by really large centra. It's one of the most abundant groups of fish in the Bissekty Formation. Nearly 40% of the centra in Martin's sample were this elopiform. It looks very similar to paratarpon, and the question will be how close it is. We have material I think will be useful in sorting that out. This skull was recently prepared, collected a long time ago, but only recently realized as being a significant specimen and recently prepared. My plan is to CT scan it and fully segment it and describe it completely. But even as it is, you can see the lower jaw has features that allows isolated lower jaws, both the complete lower jaw on the upper, your right, and partial lower jaw on the lower picture. I can compare these with the Bissekty Formation. There is a minor difference in that the row of pores, sensory canal pores on the Bissekty Formation is higher up than in the North American one. Not sure the significance of that at this point, but with the material available, I think it'll be possible to work out exactly how closely related the Bissekty Formation and Dinosaur Park Formation elopiforms are. Another interesting centrum is this one. Again, all we know is centra. It shows a couple of features that are distinctive, and one is the neural arch is very long, the centra is elongate, the neural arch is long, the parapophysis is long and slopes ventrolaterally. And this set of features is present in eels. So I've questionably said this might be an eel. If that's true, it'll be significant because it's the first evidence for eels in the Cretaceous. But we need a lot more material to show that this is really the case, as well as a full review of the centra of living eels. It's present in both Asia and North America. It's in the Bissekty Formation, but in North America, it only occurs in the very end of the Cretaceous, in the late Maastrichtian. This is one that really stood out to me as being indistinguishable between the North American and Asian specimens. This is centra from the mid-dorsal region of a fish, Coriops, which is an osteoglossomorph. And again, it's one that you could put the two in the same assemblage and can't tell them apart. Diplomistus is a herring well known from the Green River Formation. It was isolated centra. These were studied by Julien Devey, and he was able to identify isolated centra. So we can identify it in microvertebrate sites, and it's pretty widespread in the Cretaceous and present basically throughout the Cretaceous in North America, as well as being in both the Cenomanian and Turonian in Asia. Another sort of problematic taxon, I think, again, is a herring based on its similarity to one from North America called horseshoeichthyes, again, present in both Asia and North America and virtually identical. Again, virtually identical centra. These, based on comparison with recent fish, I'm identifying as a close relative of the living herrings. And in North America, it's present in Dinosaur Park, in the Kaparwitz Formation, in the Late Campanian, and then in the Turonian in Uzbekistan. Another one that Julien Devey worked on was another fish known from the Green River called Notagonius. And again, he was able to identify isolated elements. So this lower one is Notagonius from Dinosaur Park that can be identified by comparison with the articulated specimen. And the upper one is centrum from Uzbekistan that I'm identifying as the same thing. Again, a taxon that is virtually identical in both of these places. Can't say too much about this. It's widespread in North America. We think it's related to Notagonius, but we don't know anything about its geography other than it's in North America and Uzbekistan. One of the most unexpected occurrences in Uzbekistan was this fish called Primuluchara, a characiform. Characiforms are a living group of fish present in South America. They're represented by things like the piranha and the tetras that you know from aquarium fish. And it was identified by Mike Newbury, this jaw from Dinosaur Park, based on the symphysial hinge. There's a very distinctive pattern of ridges that radiate, sort of wedge-shaped ridges that radiate out from a point on the lateral side of the symphysis. and the two sides of the jaws interlock with these hinges. There are other fish that have hinges, and Codus is one that is well known to have hinges, but the shape is very different. So that it's not just the presence of this hinge Mike was using for identifying it as a characiform, but also the shape and the orientation, the recognition of them. And this is a comparison of the hinge region of the Uzbekistan specimen with the two that Mike illustrated to document its presence in Uzbekistan. Characiforms, I said, are stereophycins, and one of the features of their group is having a modified vertebra at the anterior of the column that serves as an ear. Essentially, it's got a series of ossicles that transmit vibrations from the swim bladder to the brain case. And we can look at these in recent fish. If we're correct in identifying these jaws as characiform, that fish should have a Weberian apparatus as well. So we can look at these in recent fish and then digitally segment out and look at just the centra, and then compare these with the isolated centra and identify centra of the characiform centra from the Late Cretaceous. And these are ones from Dinosaur Park. And looking at the Uzbekistan, again, we're seeing the same kind of centra. We're using this as evidence that supporting the lower jaws, that this is a characiform by the presence of these centra that form part of the Weberian apparatus. The presence of a characiform in North America, I've indicated in the initial chart that it's present in the Turonian of North America. That's based on unpublished evidence that comes out of a thesis of Aaron Kilmery, who some of you know as a former technician of the Tyrrell Museum here. So Aaron did his master's on this locality, which is a Turonian locality in Manitoba of limestone that he acid prepared and got a pretty interesting fauna of fish from the locality. And one of the specimens that he got was this jaw, which has the same kind of hinge as the ones from the Late Cretaceous and from Uzbekistan. So that's indicating its presence in the Turonian in North America. So there's a lot more work to be done on this, but I'm using this as evidence that it was present in North America. And this adds to the evidence that there was a dispersal of fish from Gondwana into North America at the Turonian. Their distribution today is clearly South American or Southern Hemisphere. They're present in South America and Africa. And fossil record is also consistent with their presence in the Southern Hemisphere. So our record in North America is the first Cretaceous record outside of this Gondwana fauna, which caused a lot of people to question it, which is why I'm spending a lot of time providing argument that it is valid. So it's everybody who works on characiforms agrees that if they're in North America, they must have dispersed there from the Southern Hemisphere. So again, evidence for a faunal change in the Turonian that involves dispersal from Southern Hemisphere into Laurasia. And when Mike Newbury and a subsequent author, Steve Wick, looked at this question of dispersal, the routes they considered were somehow from South America or from Europe into Laurasia. Finding their presence in Uzbekistan opens another possible route in that perhaps they went from Africa, Europe into Asia, and then into North America. We'll probably never get the full answer to this question, but clearly their presence in North America is clearly a result of dispersal. A couple more groups. Acanthamorphs, again, are abundant in the Uzbekistan assemblage, well represented by centra and spines. Unfortunately, we don't have any jaws, so we can't say anything about their relationships. However, it's striking in that the elements are big and they're relatively abundant, whereas in North America, they don't occur until the subsequent Coniacean period. And in that period, they're extremely rare and extremely small, so that they're clearly playing different roles in the ecosystem. Esochids are, the group that includes like the pike. In North America, they're known by jaws and centra from the Santonian through the Late Cretaceous. I'm identifying them in Uzbekistan based on this upper centra, which has the same general shape as centra from that we know from pike from North America. We don't yet have jaws, So again, how close they're related is not known. And that's why I'm encouraging Martin to continue to work in this locality. Finally, another taxon that has distinctive centra, smooth sides, long articular surfaces for the neural arch and parapophyses, but we have no idea what it is. So that's the general cast of characters when you're looking at fish from Uzbekistan. What I want to do now is look at patterns of similarity. I've indicated timing of occurrence. When we group them together and look at similar patterns, we can recognize a number of patterns. There's a core of the 18 taxa that I'm recognizing in Uzbekistan. There's a core 6 that are present both in the Cenomanian and the Turonian. So these are like the ones that are carrying on from the Cenomanian through this faunal event of the Turonian, into the Turonian. And so that's 30%. So 70% of the fauna in Uzbekistan are not known to occur in Asia prior to the Turonian. So this is really, it will certainly become less with more work, but it's certainly a major faunal change. So of those 70%, one of them is endemic, only one. So that's clearly indicating that there's strong connections between Asia and North America. There's 3 cases of taxa that occur in North America before they occur in Asia. So these are candidates for consideration of dispersal from North America into Asia. The problem is these are both all represented by small elements that are not well documented in the Bissekty Formation. So I wouldn't put a lot of weight on this it's certainly a hypothesis to be tested, but I would not be surprised if it fails the test. Again, there's a number of taxa that occur at the same time in both Asia and North America. And at least 2 of these are taxa that we think are pretty clearly dispersed from Gondwana. So this is the evidence that the faunal change that's happening is a global one. It's not a result of local changes in these areas. And then there's 5 taxa that occur both in Asia and North America, but don't occur in North America until younger time periods. So the acanthamorphs first occur in the Coniacean, the subsequent stage of the Late Cretaceous. Issochids first occur in the Santonian, the Milk River Formation of southern Alberta in North America. The Notagonius in North America first occurs in the early Campanian and is actually represented by a specimen that was collected from the Diamond Nines in the Northwest Territories. Clupeiforms, these centra that look like those of modern herring, are present in North America only in the Campanian. And then this taxon that may be an eel is only present in North America in the late Maastrichtian. So based on these patterns, we can come up with a number of hypotheses and suggestions. And one that seems pretty solid is that there's a major faunal change between the Cenomanian and the Turonian, and that this changes were similar in North America and Asia. And further, we're pretty confident based on the sample we've got that this change is a result of dispersal of taxa from Gondwana into Laurasia. I've talked about 3. Yushuki, who is a student from Japan who is visiting, has another one in he's also studying fish from the Bissekty Formation, and he has an addition to this of something related to Melvius that he's got evidence for in the Bissekty. So this idea that there is faunal change that involved dispersal from Gondwana into North America is going to get stronger with the work he's doing now too. The other aspect of the pattern is that the fish present in the Turonian are occurring to North America in a stepwise pattern. So this implies that there is dispersal from Asia to North America, but it's happening at different times. Again, this is less solidly supported because of we need larger sample size from various areas. So this is a hypothesis to be tested, but it's of interest that it's similar to the pattern seen in turtles, so that it's suggesting that there are times when interchange can happen and times when it couldn't. The obvious explanation for this is global temperature change, climate change. Because the connection between Asia and North America was at very high latitude, that there was times when these fish could live in that high latitude and times they couldn't, which meant that there's times they could disperse from Asia and North America and times they couldn't. So where do we go from here? These are the projects that I'm planning on looking at to continue this. And one is looking more closely at the Cenomanian localities in Uzbekistan, trying to get a better handle on what's there in the very base of the Cretaceous, and especially hoping to find localities that can be screen washed. This locality in the Turonian of Manitoba is very interesting in having a characiform. So I will be working with Aaron to try and get a better handle on the diversity and relationships of the fish there. And the other aspect of the study that can be done leading to a better understanding of this pattern is looking at individual taxa. I've talked about paratarpon as one that's ripe for study. The acanthamorphs, there's material available to look at acanthamorphs that'll give a better idea of the diversity of acanthamorphs in North America. And we'll be working on that with Ellison Murray from the University of Alberta. And also the characiform. We have characiforms from a number of localities and a number of time periods. And there's enough material to take a close look at them and work out, see what we can say about their relationships with the modern groups and the diversity within the Late Cretaceous. So that's a summary of where I've come and where I'm planning to go, essentially how I plan to spend my retirement. And I want to end by acknowledge a number of people, especially collection staff, because I'm working largely on specimens in collections, and maintaining those collections and making them available for study is key for the paleontological work and others are doing. So I want to acknowledge collection staff and also all the colleagues I've worked with on the research project connected to this. And part of this has been a result of working with CT scans done at the synchrotron. So the beamline scientists that work at those synchrotrons and help us out are very, very patient with somebody who can be a little technologically challenged. So I need to recognize their contribution. So with that, I thank you and would be happy to answer questions.
So do the various fish, do they inform any like in paleo-environmental reconstructions? And does that give you any clues about the way they would have dispersed?
Let's see, the fish fauna, the one that's most relevant is the one from Alaska. And because that's saying what's in Alaska at that time. Because Alaska is, that high latitude is going to be the barrier. So, and the study we had is of a period when it was relatively cool climate. And there's a very distinctive fauna dominated by esocids with the salmonids and missing things like gar and amiids, a lot of the fish we've come to think of as typical of the Cretaceous. So in terms of the fish biology, what we know of fish biology, drawing inferences to dispersal, that's our best source of information.
Don, are there very good collections out of the other East Central Asian countries like Kazakhstan, for example?
Kazakhstan is of interest in having a Santonian locality. It was not worked by the Urbach expeditions, so there's no collections that are available. But I believe that's one of the areas Martin's planning on looking at, and that would be a significant addition, because then you'd be starting to look at questions of whether there was dispersal of North American taxa into Asia. I'm sure it'll come. I'm not sure it'll come in the next 2 years, but so Kazakhstan would be an area of interest. The question is the fossil fish record of high enough resolution to show whether fish rates of dispersal versus that of terrestrial taxa? No. Our record of fossil fish in Asia is much too spotty to look at rates of dispersal. We've got a pretty dense record in North America, but not a dense enough record in Asia to talk about rates and certainly compared to mammals. The mammal record is much denser than the fish record. That's a long way down the road.
Characiforms today are freshwater,
Yep.
and I don't doubt that the characiforms you showed in the fossil record from Alberta are in fact characiforms.
Yep.
So how do you get freshwater fish in South America to North America when you showed a series of islands? Like, what's the mechanism for getting them from freshwater and going island to island, having to swim through seawater? Or
That's a problem. And that's why the identification of that as a characiform was really looked at closely. There's two approaches that I can conceive. There's a study looking at fossil fish and fossil freshwater fish that made the point that maybe this marine barrier is not as strong as we think it is, and that the barrier is more ecological than it is physical. So that's one possibility, that they were actually a marine, they lived in marine, as part of this faunal this climate change that's happening in the Turonian that really had a big impact on both marine and non-marine. Another possibility is that at least in Manitoba, there's interpretation in that sequence of beds that at least some of them were stratified, and they were either there was a basically layer of fresh water on top of a marine layer, which is sort of the analog would be the mouth of the Amazon, where fresh water extends well out to sea. That specific locality really does have a depauperate invertebrate fauna. So there's ecologically, it's something unusual. But to answer your question, yeah, the impact of dispersal from, to disperse from Gondwana to North America at that time period, there's got to be some kind of crossing marine water. And for characiforms, this is a big question.
My question is, what are the fish records like from this time period in Gondwana, in like South America and Africa?
The fish record from Gondwana at this time?
Yeah.
The best record is Brazil. Name escapes me right now. But there's a diverse assemblage preserved as three-dimensional nodules. And in Africa, there's in Morocco, there's a record of freshwater fish from the Cenomanian. Santana, yeah, that's it. Thanks.
So I'm interested in our acanthomorphs. You mentioned, I think, was it Coniacian was your first in Alberta, your first center on
America.
And in considering with our acanthomorphs, we also have some marine acanthomorphs like the Xenilian from the marine formation, which I believe is a lot earlier.
Yeah.
I'm interested, have you looked at Xenilian, and do we have centra from the fish scale zone? And how do those compare with our other freshwater taxa? And could that be like an independent freshwater marine transition in North America, or what do you think, especially with acanthomorphs where we know that they're in both freshwater and marine?
Yeah, almost certainly. That's one where it's to answer your first part of the question, no, I haven't looked at the material yet. I plan to. There's a good record of isolated acanthomorphs in this Montana locality, and I want to see if they can be identified as any of the things we know from the marine. It's highly probable that they entered freshwater conditions independently in these two regions. For those kinds of questions, we need a better understanding of the phylogenetic relationships. And there's faunal changes in acanthomorphs later in the Cretaceous, so in the Maastrichtian, more derived acanthomorphs appearing. So there's going to be a story there somehow. We don't have it yet. We just know that this is what we got. What might be the reason for the fossil records being more dense in North America than in Asia? Why might that speak to or signify? it's a reflection of the environments, well, a number of things, but partly the environments in these different areas, so that in part of the time you're dealing with marine and part of the time you're dealing with freshwater. So there's a limited number of freshwater localities in this region of Asia, in Uzbekistan. There's a lot in like Mongolia and China, but these are arid. So the kind of fluvial localities I'm interested in are most developed in Asia, in Middle Asia, Central Asia. And there's only a few specific time periods presented. In North America, the record is denser because of the geography. There was a North America, or the part of North America we're in was a long, narrow peninsula, essentially, that extended from Alaska down through what is now the Rocky Mountains to Mexico. The shore of the inland sea came and went, covering part of the floodplain on the east side of this peninsula. And every time it went, there was non-marine deposits deposited, which is why there's a series of non-marine deposits in North America. When you look at North America as a whole, including Utah and Canada and Montana and everywhere. So that's why we have a number of time periods presented in North America. What other sites across the former Soviet Union may yield further information on the Cenomanian Turonian transition? The material I talked to came from one specific site in one formation, a locality called Characiform in the Bissekty Formation. There's other areas of exposure of that formation, and Yusuke is working on material, so we'll come up with more material from some of these other areas, I'm sure. There's other time periods like in Kazakhstan that are known to be have freshwater fish assemblages. So yeah, there's more there. Somebody's got some fieldwork possibilities ahead of them.
Thank you very much for that wonderful talk, Dr. Brinkman, and please accept this small token of our appreciation for your talk today.
Thank you.